Hello everyone, this is Larry Rothfield, and this will be the first lecture in a two-part series on coronaviruses. This series is designed for non-specialists. Nevertheless, the science has not been dumbed down or watered down, although I have omitted a number of details that I think are unnecessary for a true understanding of the basic mechanisms involved in how coronaviruses cause disease, and I have simplified some of the vocabulary. There are a large number of human coronaviruses. For example, in this country, it has been estimated that approximately 15% of colds and upper respiratory infections are due to coronaviruses. But the coronaviruses that concern us are the three viruses shown on the right that were responsible for the epidemics and pandemics of this century. These viruses fall in two classes: the MERS group for Mideast Respiratory Syndrome virus, and the SARS group for Southeast Asia or Severe Acute Respiratory Syndrome virus. The COVID-19 virus is a member of the SARS group, and the genes in the COVID-19 agent show 95% identity with the genes of the SARS virus that caused the 2003 epidemic. All three viruses have caused large numbers of deaths, and for the present pandemic, we're still counting. In the United States, for example, the number of deaths has now passed the 80,000 mark and will surely be significantly higher within the months to come. In any case, no matter how you cut this, these are very deadly viruses. The mortality rates shown here are almost surely an overstatement, especially during the two preceding epidemics. The number of patients with mild disease, with mild symptoms or no symptoms, was almost surely seriously undercounted, so that those numbers are probably quite high. The mortality rate for the COVID-19 pandemic has been in the 1 to 2% range, and that's probably about correct, although that may also be lower once countries like the United States make widespread testing more accessible and more data becomes accessible. Now, the very sobering part of this slide is the fact that within the last 3 decades of this century, there have been 3 major outbreaks of deadly coronaviruses occurring every 8 or 9 years. Now, this strongly suggests, probably with some degree of confidence, that sometime within the next 8 to 10 years, there will be another major outbreak of one of these viruses that acquired the ability to jump from animals into humans. There's no question that if we don't act in a more timely manner to deal with these future epidemics, we're destined to have the same deadly effect and the same huge number of deaths worldwide. Coronaviruses bear a surprising resemblance to the naval mines which were used to destroy enemy ships during World War II. That's directed at those of you who may just be old enough to remember World War II. In both cases, these are spherical structures. In both cases, spikes protrude from the surface, and in both cases, the spikes are the first component of the structures to interact with the target, with an enemy ship, in the case of the naval mines, in humans, of interaction with human cells, in the case of coronaviruses. And in both cases, the initial interaction sets in train a sequence of events that leads to destruction of the target. This is an electron micrograph of a coronavirus enlarged several thousand-fold before being projected on your television screen. As you can see, there are three visible structures within the virus particle: the spikes that protrude from the cell surface, a thin membrane indicated in the thin blue line that underlies the spike layer and surrounds the interior of the sphere, the membrane layer, and in the interior, colored red in this pseudo-colored electron micrograph, is the viral chromosome that makes up its genetic material and the several proteins required for replication of the viral chromosome that are also present in this region. In coronaviruses, the genetic material is RNA instead of DNA, as it is in all cells, but this need not concern us, because for all intents and purposes, RNA and DNA genomes behave similarly within the cell. On this slide, we compare the number of genes that are present in cells and viruses, and what's immediately apparent is there's an enormous discrepancy in the number of genes present in cells as compared with viruses. In all cells, from human cells down to simple single-celled organisms, bacteria, the cells contain anywhere from 4,000 to 20,000 individual genes. In stark contrast to this, the number of genes in coronaviruses is 20, and in even smaller viruses such as the poliovirus, the number of genes can be as low as 5. And the question we will return to is how viruses with these vanishingly small numbers of genes can manage to survive in nature. So let's discuss why this is important. This is important because the major function of genes that we know about is to code for proteins. One gene produces one protein. And since proteins are responsible for everything that happens within the human cell, ranging from cell division, chromosome replication, metabolism, and so forth, it's not surprising that the genes that human cells, cells of complex organisms, contain such a large number of genes. The question, of course, again, will be how do viruses survive with their small complement of genes? So to help answer this, let's take a look at the types of proteins that are produced by the 20 coronavirus genes. These can be divided into two groups, the structural proteins and non-structural proteins. So the structural proteins include the spike proteins, one or two membrane proteins, and perhaps other structural proteins of the virus particle. The non-structural proteins include a number of proteins required for replication of the viral chromosome, replication of the viral genome. These are associated with the RNA in the center of the cell. About 6 proteins appear to be responsible for pathogenesis, that is, for the ability of the virus to cause disease. And these, of course, are of most interest to us. The biochemical functions of 2 of these proteins is known, and the functions of the other 4 is completely unknown. And it's quite clear that this is a large gap in our knowledge and has to be a major target of research in this important area. Let's take a look at the structure of cells and see how this compares with the structure of viruses. All cells, and cells comprise everything within the human body, all cells have a similar structure. All cells are surrounded by a membrane indicated in orange in this cartoon, which surrounds the interior structures of the cell. Within the interior of the cell in humans and animals, there are several small structures that are visible, such as the nucleus, indicated in N here, which contains the 46 chromosomes, human chromosomes. In the cytoplasm of the cell, there are hundreds of additional proteins, which are required for carrying out all of the many functions that keep cells alive and that permit them to function within the human body. But the most important part of the cell, for us, will be the structures on the outside surface of the cell, the receptors which protrude from the cell membrane. As indicated in this cartoon, there are many different species, many different classes of receptors. What is the function of these receptors? The function of these receptors is to act as binding sites, which is recognized by a number of molecules which must interact with the cell, in many cases gain entry into the cell interior, in order to maintain the life and health of the cell, and by extension, maintain the life of the human body. Now, the blow-up below indicates the important part of these receptors for our purpose, and these will be quite important later when we discuss how coronaviruses cause disease. Each receptor protein has on its surface a small pocket or cavity that differs in structure and size from receptor class to receptor class. These differences in structure of the receptors makes it possible for each of them to act as a binding site for a different partner. So the receptor for insulin, for example, will not be recognized by other hormones or by the sugar glucose, whereas the receptor for glucose will not recognize other sugars or other small molecules. And it's this difference in receptor specificity of the surface receptors that will be exploited by the virus, as we will discuss later. There are different classes of cell receptors. One class recognizes nutrients, for example, sugars and amino acids, that are required to provide the energy needs of the cell and to act as building blocks for the manufacture of proteins and DNA and other essential elements of the cell. There are receptors for hormones, which are in some cases essential to life, such as insulin, in other cases important for function but not essential. For example, estrogens, the estrogen receptor, as you probably know, is the target for treatment with drugs that play important roles in treatment of breast cancer. And there are receptors that are responsible for interacting with other cells, that is, responsible for cell-cell interactions. These are obviously of enormous importance, essential importance, in the formation of the structures that differentiate organs such as the kidney from other organs such as the liver. Now, how do cells differ structurally from viruses? Well, at first glance, they're really quite similar. Both of them are membrane-bounded structures containing the lipid-containing membrane that surrounds the entire interior of the cell. And in both cases, the surface have receptor proteins protruding from the surface. The many receptor proteins on the surface of animal cells that I've just described, and the single spike proteins on the surface of viruses. Well, how do viruses differ from cells then? Well, as I mentioned before, the key difference, of course, is in the number of genes in cells and in viruses. Cells containing thousands and thousands of genes, viruses containing perhaps between 5 and 20 genes. And this results in the fact that cells can independently synthesize their own proteins, replicate their chromosomes, that is, replicate their genetic material, their genomes, and reproduce themselves. In contrast, viruses can do none of these things. Well, if this is so, how can viruses survive in nature? And the answer is that they survive by utilizing the cell's machinery to carry out all the essential tasks required for their survival. For example, they use the cell's protein synthetic machinery to make proteins coded for by each of the viral genes. So let's look at two questions. First, if viruses have to get into cells to use the cell's machinery to make their proteins and reproduce the viruses, how do viruses gain entry into cells? And secondly, what do they do when they get there? Let's first discuss how viruses gain entry into cells. As I mentioned a few moments ago, the cell receptors have highly specific binding sites on their surface. And viruses, the first stage in viral entry is to bind to one of these binding sites. To accomplish this, the viral spike protein contains at its tip a site that mimics the normal binding partner of one or another class of receptors. In this way, the virus recognizes the specific receptor and binds to the surface in the first step, step 1, of the sequence of events that will lead ultimately to viral reproduction within the cell and cell killing or cell damage. The second stage is entry into the cytoplasm. And what's essential for the virus to get into the cytoplasm is the viral chromosomes, since it's the chromosome of the virus that contains the genes that will code for viral proteins, which will be needed to make up new virus particles when they reproduce within the cell interior. Now, I've simplified this by indicating that the only part of the virus that enters the interior of the cell is the viral chromosome. In fact, that is true for a number of viral types in which only the genome, only the chromosomes are injected into the interior of the cell. For membrane-bounded viruses, such as coronaviruses, the mechanism is more complicated. But for our purposes, it really doesn't matter. The important thing is to get the genetic material inside so they can make viral proteins that are going to be used to assemble into the complete virus within the interior of the cell. The third stage in the series of events that leads to viral reproduction is to replicate their genetic material, to make duplicate copies of the chromosome. The viruses accomplish this within the cell so that within a relatively short time, there are many, many copies, identical copies of the viral chromosome. Each one of these copies containing the 20 genes of the virus capable of making or coding for the 20 proteins that are part of the mature virus particle. To make these proteins, the viruses use the protein synthesizing machinery of the cell. So at this point, the viruses will have within the cell the viral chromosome and the viral proteins, everything that will be needed to assemble a mature virus. And this leads to the fourth stage in the viral reproduction cycle, namely assembly of the complete virus. The viruses there self-assemble within the cytoplasm, which then contains copies of mature virus particles within the cell. And this leads to the final event that occurs, namely cell death. This occurs by the continued reproduction of the virus, in which the large number of virus particles within the cell appropriate all of the cell's machinery, ending up ultimately destroying the cell and releasing large numbers of progeny viruses into the surrounding tissues in the case of human infection, where each of these virus particles can now seek out another cell to infect, reproduce, and kill. After killing the infected cell, the viral progeny that have left the safe haven of the cell interior must find and infect another target cell in order to continue the cycles of cell destruction that lead to disease in the infected host. Let us examine this for a moment, because it provides a period of vulnerability for the infectious virus. Now, reinfection, infection of a new cell, requires the productive interaction of each virus particle with a new host cell. And this can only occur by the random collision of the virus with a target cell. Furthermore, the collision must be with one specific receptor amongst the large number of other receptors on the surface of the target cell. This requirement for a highly specific collision event means that only a small number of virus particles are likely to succeed in rapidly and productively interacting and reproducing within a new target cell. This relatively inefficient reinfection process provides a relatively long period during which the released virus will be vulnerable to host defenses or drugs. And this is something we will discuss in the second lecture. Well, we've come to the end of the first lecture in this two-part series. If any of you have any questions, please don't hesitate to contact me, and I will do my best to get back to you. So let's move on now to the second and last lecture in this series. This will be the second lecture in this two-part series on coronaviruses. From Lecture 1, we drew the following conclusions. First, viruses are obligate intracellular parasites unable to survive in the absence of cells. Secondly, the viruses gain entry into the cell by hijacking the normal cell surface receptors. Thirdly, the viruses then utilize the cell's normal machinery to reproduce the virus within the cell. And finally, after killing the host cell, the viruses must find another cell to infect before they are themselves inactivated within the environment. In this lecture, we will try to answer two general questions. First, how do coronaviruses cause disease? And secondly, how can we prevent and treat the disease? We'll begin by briefly discussing how pandemics begin. Genetic evidence indicates that for all three coronaviruses, the human viruses evolved from a common ancestor in bats, where the virus apparently does not cause serious disease. Because humans rarely have contact with bats or bat droppings, it's believed that for all three viruses, an intermediate animal host is interposed, passing from bats to the intermediate host, where the virus mutates and gains the ability to infect humans. For the Mideast Respiratory Syndrome virus, MERS, there's reasonably good evidence from genetics that camels are the intermediate host, and the virus in fact did emerge in Saudi Arabia. For the other two viruses, it's not known what the immediate host is. Following inhalation, the virus attacks the pharynx and upper respiratory tract, leading to a sore throat, an extreme sore throat, and upper respiratory infection. Virus particles that are released from damaged cells in the throat and upper respiratory tract then pass down the bronchial tree into the lungs, thereby adding significantly to the viral load that reaches the lungs. As shown in the cartoon at the lower left, the bronchial tree is terminated by hundreds of air sacs which are scattered throughout the lung. The air sac is an air-filled cavity, indicated in white, surrounded by a layer of lining cells, indicated in blue. As we discussed in our last lecture, invasion of the lining cells by viruses within the cavity lead to cell destruction and release of virus. When sufficient air sacs have been affected in this way, the air sac damage that's resulted can interfere significantly with the amount of oxygen that the lungs can take up and transfer into the circulatory system. And this can be part of the cause of disease caused by COVID-19. Now the host attempts to respond to this viral onslaught. The initial host response to foreign invasion is a local response in the infected tissue, the innate immune response. This local response has two characteristics. First, it is immediate, beginning as soon as the viral particle reaches the infected tissue, in this case the lung. Secondly, it is nonspecific, meaning that it acts equally on bacteria and viruses and does not discriminate between different classes of viral invaders. The way in which the local innate immune response deals with invading viruses is quite simple. Virus particles that arrive within the lung sacs enter the lining cells of the air sac, resulting in release of progeny virus into the surrounding tissue. The viruses then signal to scavenger cells located elsewhere in the lungs, stimulating them to migrate to the site of cell damage, where the scavenger cells interact nonspecifically with the invading viral particles as indicated by the white arrow. The surface-bound virus particle is then engulfed into the interior of the scavenger cell, where it's present within a membrane-bounded vacuole. And this is followed by the last stage in destruction of the viral particle, in which the virus is digested within the vacuole by host cell digestive enzymes. Now, in addition to the local innate immunity response that we've just discussed, human hosts have a second and even more important defense mechanism to protect against invaders, and that is the systemic antibody-mediated immune response. The antibody-mediated and the innate immunity response differ in two important ways. Whereas the local response is nonspecific, the antibody-mediated response is highly specific. Each antibody will recognize and protect against only a single, highly specific class of viruses. And secondly, whereas the local response is immediate, the systemic response is delayed, reflecting the time it takes for the body to generate the very highly specific antibody directed against only one of hundreds of thousands of possible invaders. Now, how does this very important antibody-mediated host defense machinery actually work? Virus particles are released from damaged cells into the circulation, where they signal antibody-producing cells located elsewhere in the body, primarily in the lymph nodes and spleen. In response to this viral stimulation, antibody-producing cells generate highly specific antibodies that are released into the circulation. What are antibodies? Antibodies are proteins containing highly specific binding sites on their surface. In the case of coronaviruses, antibodies almost exclusively recognize the surface proteins that are protruding from the surface of the viral particle. As a result, when circulating antibodies encounter the invading viral particle, they bind tightly to the surface of the virus, as indicated by the arrowhead. This marks the virus particle for rapid destruction by the host. The destruction of the virus antibody complexes is much more efficient than destruction of free virus by the innate immune response. As a result, the innate response, the immediate immune response, can deal effectively with relatively small numbers of virus particles. The innate response can abort infections with smaller viral loads or attempt to hold the infection in check with higher viral loads until antibodies can ride to the rescue. Let us consider for a moment what's involved following the first exposure of a human host to a virus it has never before encountered. The antibody-producing machinery of the cell must learn to make a highly specific antibody protein molecule, which will recognize, for example, the spike protein of the COVID-19 virus, which it has never before encountered. Now, when you consider the thousands, or actually the hundreds of thousands of possible stimuli to the antibody response mechanism, it's not surprising that learning to make this new specific antibody takes some time. And this explains the fact that antibody responses to new stimuli, such as a virus to which the host has never before been exposed, takes anywhere from 1 to 2 weeks, with significant levels of antibody not appearing until the latter part of this period. Now, the situation is quite different with subsequent exposures because of the fact that the body has immunological memory for exposures that they've experienced before. This immunological memory is, in fact, a few antibody-producing cells which remain alive in the host for decades or often a lifetime for some viruses, so that a subsequent exposure to the virus results in a much more rapid antibody response, usually in the 2 to 3 day range, and moreover, and quite important, subsequent challenges of this type often lead to much higher concentrations of antibody in the circulation. Now, there are 3 things that are worth remembering here. First, as I've already mentioned, is there is a delay, a significant delay, following first exposure to the virus before antibodies appear. And during this time, with a virus such as the COVID-19 virus, the host can be badly damaged or killed. Secondly, it's not known whether in fact the antibody response to COVID-19 will or will not be protective. With most viruses, the antibody response is protective, but this is not universally true. And thirdly, it's not known for the COVID-19 virus how long immunological memory will be effective. That is, for how long a period subsequent exposures will stimulate a new outburst of antibody formation, and this varies from virus to virus. Let us return now to the question of how do coronaviruses cause disease. As we discussed earlier, the virus itself can cause severe damage to the pulmonary air sacs, resulting in interference with oxygen uptake. However, this is not the major cause of severe disease leading to death in these patients. Instead, severe disease caused by coronaviruses leading to death results from a host response mechanism reversing itself and beginning to attack the tissues and organs of the infected patient. So the mechanisms designed to attack the virus instead attack the host itself. And this occurs through something called a cytokine storm. So let us examine what's involved in a cytokine storm. The cytokine storm begins with release of virus particles from infected cells. The released particles then signal other cells within the lung to synthesize cytokines. Now, what are cytokines? Cytokines are small molecules that regulate various aspects of the immune response. The released cytokines in the lung in turn signal scavenger cells located elsewhere in the lung to migrate to the site of cell damage, where they interact with virus particles and destroy them in the mechanism we've already discussed. In the case of highly virulent viruses, such as the coronaviruses, for example, the number of virus particles released is too large to be destroyed by the initial wave of scavenger cell migration. As a result, the scavenger cells within the lung elaborate more cytokines, and these stimulate the migration of more scavenger cells to the site of cell damage. This vicious cycle, scavenger cells migrating to the site of cell damage, releasing more cytokines, provoking migration of more scavenger cells, and so forth. This large excess of scavenger cells now, in addition to attacking virus particles, begins to attack normal cell structures themselves, resulting in extensive damage and destruction to cells of the pulmonary air sacs. At the same time, the large number of cytokines that have accumulated in the tissue enter the bloodstream, where they're carried to downstream organs, and within these other organs of the body, this same vicious cycle is repeated with disastrous effects. This results in massive failure of many of the body organs, lung especially, kidney, liver, and other body organs. The damage to the lung is especially dangerous and is the proximate cause of death in most patients. Damaged capillaries result in a leakage of fluid into the air sacs of the lung, which very rapidly, often within a question of hours, interferes sufficiently with the ability of the lung to transfer oxygen into the circulation at a level sufficient to sustain life. We all know from reading the newspapers, mechanical ventilators are used clinically in an often futile attempt to push enough oxygen into the circulation to maintain life until the body's defense mechanisms can rid the body of the virus and repair the damaged tissue. You'll recall that human hosts defend against viral infection in two ways: by utilizing the immediate innate immunity system or the acquired antibody-dependent immunity system. In response, viruses have evolved mechanisms to evade both of these host defenses. We've already heard about how viruses counteract the innate immunity response by generating large numbers of viral particles that overwhelm the response, making it possible for cytokine storms to cause disease and death in infected patients. To counteract the antibody-mediated immunity response, viruses resort to mutations. Mutations are changes in genes that result in changes in the structure of their protein products. Again, you'll recall that the viral spike protein is a protein that contains the recognition site used by antibodies in their attempt to destroy the viral invaders. Therefore, mutations within the viral spike protein make the virus invisible to antibodies, thereby making it resistant to antibody-dependent killing. This is well exemplified by influenza virus, which you'll recall each winter requires a new vaccine because mutation of influenza after the preceding influenza season has generated a new virus that is no longer recognized by last year's antibody. There are two additional host defenses against viral infection and viral-caused diseases. The first is mechanisms that prevent either entry of the virus into the human host or its establishment of disease, and second, mechanisms that treat the disease once it has occurred. There are several well-known ways of preventing viral infection. These include common sense activities, contact tracing, social distancing, use of face masks, the use of high-efficiency filters capable of filtering out virus particles in internal air recirculating systems. The virus can also be killed before entry locally. The use of detergents kills viruses by dissolving the fat-containing membrane that surrounds the virus particle. Ultraviolet light kills viruses by destroying the genetic material, either DNA or, in the case of coronaviruses, RNA. And finally, the most effective and most important way of preventing infection by virulent viruses is vaccine development. This is well underway, as you know from reading newspapers, and hopefully should be available by sometime next winter, although it's always very difficult to predict these things. There are two current modes of treatment available for virally infected patients: immunotherapy, that is, the use of antibodies, and drug therapy. When we speak about immunotherapy, we're speaking about infusion of either immune serum obtained from the blood of patients who have recently recovered from treatment with coronaviruses and whose blood contains a high level of antibodies, or the infusion of purified antibody. Now, serum or plasma are essentially the same thing, namely the material that's obtained after removing white and red blood cells from blood by centrifugation. There are several things that must be considered in providing immunotherapy. First, the antibody must be protective. In most cases of viral diseases, antibodies in blood from recovered patients usually is protective, but this is not invariably so. Second, in the case of infusion of immune serum, the serum which has been obtained from patients who have recently recovered from the virus must be shown to be virus-free. Luckily, tests for the presence of virus are very sensitive and are very reliable, so this should not be a problem. Third, again, the use of infusion of immune serum into patients with the disease, if one aims to get the same level of antibody in the recipient as was present in the blood of the donor, plasma must be pooled from several patients in order to treat one recipient. This is a serious obstacle to using this approach on a widespread basis. I've read in the newspaper that in current attempts, some people are using 1 unit of plasma from a single patient to treat several recipients, and we'll just have to see if that works. Now finally, the antibody must be of human origin. What do I mean by this? Well, in the early years of the last century, people began to try immune serum to treat patients with bacterial diseases. Since it was difficult to obtain large amounts of serum from patients, animals were immunized with the bacteria, and their blood, which contained high levels of antibody, were used to prepare serum for treatment. This had some success. Unfortunately, it was fairly quickly found that the infused antibodies, if made in animals, themselves induced serious illness in the recipients, so that this approach, the use of animal blood to obtain immune serum, cannot be used in humans. More recently, it's been possible to generate specific antiviral antibodies in the laboratory using cloned human cells, human antibody-producing cells. This approach has not been used or not been successful, to my knowledge, in the case of viral diseases, but it has been very successful in treatment of other types of diseases. These antibodies, called monoclonal antibodies, since they're generated from clones of human antibody-producing cells, may in fact prove to be extremely useful in therapy of patients with coronaviruses. There are many potential drug targets in attempts to treat viral diseases, and in fact, targeting specific stages of the viral reproductive cycle, elements 1 to 4 in this list, has proven to be successful and given useful drugs in several cases. Let's turn our attention to the first of these, drugs that act by inhibiting the binding of virus to the cell receptor. This is, as you'll recall, the first step in the reproductive cycle. You will recall that the first step in the viral reproduction cycle is interaction of the virus with one of the many protein receptors that are located on the surface of all cells. You'll also recall that binding occurs via the viral spike protein, which is a specific binding partner for the specific cell surface receptor of the recipient cell. It follows from this that drugs that interact with the virus spike protein should be effective in preventing viruses binding to the cell surface and therefore from reproducing and causing disease, as shown in this cartoon. And in fact, drugs that are effective in treatment of certain other virus diseases have been designed that act in this way, although to my knowledge, this has not yet been achieved for any coronaviruses. There is a second class of drugs, antiviral drugs, that act by preventing binding of the virus to its cell surface receptor. And these are drugs that interact themselves with the receptor. This should prevent the virus from subsequently approaching and attaching to the cell surface, as shown in the cartoon, thereby preventing the virus from infecting the cell and causing cell damage or cell destruction. This is more complicated, however, than the class of drugs that bind directly to the viral spike. And the reason this is more complicated is the fact that the receptors on the surface of cells are not placed there for the convenience of the virus, but rather because they play an important role in the life of the cell or of the human body. If, for example, a receptor plays an essential role in human life, it will be an impractical therapeutic agent for obvious reasons. However, there are a large number of receptors on the surface of cells that, although they play a useful and sometimes important role in life, are not absolutely essential. For example, other coronaviruses, and very likely the COVID-19 agent, although to my knowledge this has not yet been shown directly, These coronaviruses use as their receptor a protein which plays a role in regulation of blood pressure in humans. Since this is a non-essential function, it's conceivable that drugs that interact with the receptor may still be suitable as antiviral agents, and there's no way to tell this until the appropriate experiments are carried out. It has been difficult to develop antiviral drugs, but in recent years a number of effective drugs have been developed that treat viruses responsible for a number of viral diseases, and some of these are indicated in yellow on this slide. I'll call your attention to one especially fruitful target, stage 3 of the process, namely replication of the viral chromosome. Several drugs have now been developed that inhibit virally encoded proteins that are required for viral chromosome replication and therefore effectively block viral infections by the target virus. Some examples of these are shown here. They include the HIV virus, which causes AIDS, hepatitis B virus, and the herpes zoster virus, which causes shingles and chickenpox. It obviously would be of enormous value to have a drug which prevented or treated cytokine storms. Unfortunately, to this point, results have been very disappointing. A number of drugs have been and are being tested that are effective in treating autoimmune diseases in humans, since these presumably act by tamping down the immune response. These include chloroquine, pushed by our president. So far as I know, none of these studies have produced anything that's useful in treating COVID-19 patients, although research continues in this direction. This is a complicated business because the regulation of the human immune response is very complex. One sobering result was obtained in tests of a drug that might be expected to be effective in treating cytokine storms, in which administration of the drug to human volunteers induced severe cytokine storms. So this is a complicated business, but the value of having such a drug obviously makes research in this direction extremely important. The final possible target for drug treatment is the throat and upper respiratory tract, because sore throat and upper respiratory infection are the initial stages of COVID-19 disease in almost all patients. Because it's very likely that significant amount of viruses might be released into the upper respiratory tract at this stage of the disease, virus particles that would increase the viral load in the lung, it would be very useful if a local treatment could be developed that interfered with viability of viral particles at this early stage of the infectious process. I don't know of any work going on in this direction, but it's conceivable that it exists. So what we've learned in this series of lectures has been summarized on this slide. First, viruses are intracellular parasites unable to live in the absence of cells. Second, after killing the host cell, the virus must find another cell to infect before being inactivated by host defenses. Third, survival of the virus is a back-and-forth battle between host defense mechanisms and viral responses. Fourth, serious disease and death are usually caused by out-of-control host defense mechanisms, namely cytokine storms. And finally, in the most optimistic part of the talk, there are many attractive targets for drug design. Well, we've come to the end of this two-part series on coronaviruses. As I mentioned at the end of the first lecture, if you have any questions, please don't hesitate to contact me, and I will try to get back to you. Remember, there are never any stupid questions. There may be stupid answers, but no stupid questions. And finally, let me thank you for attending this series. I surely hope you got something out of it. And remember, please stay well.